So this is our first lecture on micro lecture on chapter 16. This is going to be in 3 parts. We're first going to do the introduction to the sense organs. We're going to kind of talk about pain, finish talking about taste, and also smell. Part 2 is going to be hearing and equilibrium, or the ear. And then part 3, the final one, is going to be light and vision, which is pretty much this vision and the eye. So let's get started on your introduction. So sensory input is vital to the integrity of personality and intellectual function. That might sound very strange, but seeing, hearing, feeling, tasting, all of these things are very, very, very important for your personality. They're also hearing, seeing, all these things can help you learn and make you smarter. Travel and everything you've probably heard, by learning new things and constantly learning is a key to a healthy life, because you need to use the cells that you've learned about this semester. So by traveling, seeing, and having the senses actually improves your mood and actually makes you smarter. Information communicated by the sense organs never comes to our conscious attention. Blood pressure, body temperature, and muscle tension. So you might not even know this, but your sense organs are bringing all these things into your body at all times. You have baroreceptors we talked about, you have chemoreceptors. Those are all types of sensory organs. And they're not the vision, they're not the taste, they're not the hearing, but they're still going to be organs within the body that help tell us about our environment that we are in right now. Sense organs initiate somatic and visceral reflexes that are indispensable for homeostasis and our survival. We've mentioned about them throughout the semester with baroreceptors, if pressure changes in cells, chemoreceptors, acid and bases, changes within the blood. These are just to try to keep our body at a certain level. 2 general properties, phasic and tonic. These are very, very, very important. And knowing which does which is very, very, very important. Phasic, generate a burst of action potentials when first stimulated, quickly adapt, and sharply reduce or stop signaling even though the stimulus continues. Smell, hair movement, cutaneous pressure. Think about that. If you are going out on a date or going out to the movies with friends or going out with friends to a bar or something, you might want to put some cologne or perfume on yourself. And when you put it on, you're like, oh, I can smell it. It smells great. And then I guarantee you, probably by the time you get in the car, you don't smell it anymore. The reason why you don't smell it anymore is because the sense of smell is a type of phasic receptor, where once you smell something new, that generates an action potential that goes straight to your brain. Your brain interprets that smell, and then after a while, it just stops even caring about that smell. And you might even apply more perfume or cologne because you don't smell it anymore. But I guarantee you the person that you meet is like, why is this person wearing so much perfume or cologne? Because you don't know that the sense of smell is phasic, so you keep adding new scents to yourself so you can keep getting that original smell. For you, going out on a date or going out with friends, that doesn't make sense. But if you think if you were in the woods and your life depended on you smelling new things, this is very important. If you have something that you're going to eat, you smell it, it smells rotten, you don't eat it. Then you can smell something upwind from you that smells like it's dangerous or musty. It could be an animal that's about to attack you. So your sense of smell always wants to be ready for new smells. And if you're going out on a date, you're probably not too worried about that, because you should be in a better, environment than something trying to get you. Hair movement and cutaneous pressure, same thing is if you put on a wetsuit that's really tight or put on something that's really tight, you only feel that pressure for a little bit and eventually it'll go away. So all of those are phasic. Tonic is different. It adapts slowly, generates nerve signals more steadily. Proprioreceptors, that for body position, muscle tension, joint motion. These things need to kind of ramp up because if your body position gets too one way, you can fall over. So it kind of ramps up slowly, and then right when you need to, you can counteract that. Muscle tension, the same thing. You don't want your muscles to, if you're just using your muscles, you don't really care about muscle tension. But the more muscle fibers you kind of recruit, the more tension that's going to be there. And then if you have almost all of your muscles fibers, being tensed, that's when you want to either let go or your muscle will fail because your body does not want to rip a muscle fiber. It wants to keep it tense. Same thing for joint motion. If you're just using your joints normally, no problem. But if you put a lot of stress on them, usually you'll feel a very intense pain right before the joint fails. And you have many different types of receptors. Thermoreceptors, heat or cold. Photoreceptors in the eye. We're going to talk about the rods and cones later on. Nociceptors for pain. We talked about nociceptors with the integumentary system or skin. Chemoreceptors respond to chemicals, including odors, tastes, and body fluids. Mechanoreceptors respond to physical deformation of cells, tissue caused by vibration, touch, pressure, stretch, tension. Certain arteries can detect changes in blood pressure. That would be a type of baroreceptor. But mechanoreceptors, mechanical. Chemoreceptors, chemical, photolight, thermoheat. Or by origin. So extero, external. Intero, internal. Proprioreceptors, body position or movement. Then distribution, general senses, widely distributed, special senses limited to the head. Vision is part 3, hearing and equilibrium is part 2, and taste and smell is what we're going to kind of look at the end of this micro lecture. So what are some of these types of receptors? So unencapsulated nerve endings. Unencapsulated means they're very, very, very close to what they need to respond to. So these dendrites not wrapped in connective tissue. So there's nothing surrounds it. You can have free nerve endings. These are for pain and temperature. And these are going to be skin and mucous membranes. And free nerve endings, why would pain and temperature, you'd want them to be free? You want them to be free and nothing around them just because pain and temperature, you want your body to respond to pain and temperature very, very, very quickly. Tactile discs have this structure that sits right on top of it, a tactile cell for light touch and texture. They're closely associated with the Merkel cells at the base of the epididymis, which we went over in AMP, or in the integumentary system in AMP1. Hair receptors too can be there. We also have dendrites wrapped in glial cells and connective tissue. We also have connective tissue enhances sensitivity or selectivity of the response. We talked about the lamellar corpuscle in the skin. So let's take a closer look at it now. The tactile or mesonar corpuscle, light touch or texture, the dermal papilla, the hairless skin. Those are the ones where we looked at the skin. They were right up top. The Krauss end bulbs, tactile in the mucous membranes. We didn't go over those. We're going to look more of those when we talk about AMP2, when we talk about the respiratory system, and we get a closer look at the mucous membranes. The one that I mentioned and showed you the picture of on the last slide, the lamellar or pressure receptor, the Pacinian corpuscle. Deep pressure, stretch, tickle, vibration. periosteum of bone, and deep dermis of the skin. These are for really changes deep in the tissue. And they do that because of that lamella or laminated structure. When they start to kind of, if you just kind of looked, there was like layers of them or laminated. When pressure pushes one layer down on another, it opens a channel, ions flow in, it sends a signal. The more of those layers that get deformed or touch each other, the more channels get open, the more signal of pressure, stretch, tickle, or vibration that you'll feel. Bulbous or Ruffinian corpuscles, tonic, heavy touch, pressure, joint movements, and skin. And that's how you get that pressure where you're wearing a tight shirt and it kind of goes away. You also have other ones that give you that slow ramp up, that if your joints start to hurt or your skin's stretching too much, then these are there to kind of tell you to get out of that situation and stop moving your joint that way, or try to stop whatever activity you're doing. Also pain, discomfort caused by tissue injury or noxious stimulation, typically leading to evasive action. Important since it helps protect us. In diabetes, you lose this in diabetic neuropathy. So you don't have these pain receptors. So usually people, they might keep their hands on, or their feet, mostly in their limbs, in places like hot. So they might have their feet up and they might have a cold pack on them, and then eventually they'll get, their tissues will stop freezing, because in diabetic neuropathy, you don't have that pain sensation anymore. So people with diabetic neuropathy need to worry about their limbs, hands and feet mostly. Nociceptors, two types providing different pain sensations, one fast, one slow. You can have somatic from skin, muscle, joints, visceral from the viscera. And remember, every time you see viscera, it's going to be around an organ or tissue in the thoracic and the abdominal cavity. Stretch, chemical, ischemia of viscera, poorly localized, which means that ischemia, lack of oxygen, which means in one location there's a poor oxygen content. Problem with that is that in ischemia, with visceral pain for this, another way to kind of look at this is you can't really determine where it's coming from. So poorly localized. Ischemia of the heart, you feel it in the arm. So if you have a heart attack, that's what that means by you can't really determine where it's coming from. Injury tissue release chemicals that stimulate pain fibers, brandykinins. You'll hear about that with different types of drugs. Histamine, prostaglandin, serotonin stimulate nociceptors. Again, drugs like Benadryl and other stuff will start to work on that. Referred pain is what I was talking about when I was talking about the heart. And referred pain is pain in the viscera, often mistakenly thought to come from the skin or other superficial sites. And it's very strange, because when these two nerve endings come together and bring stuff to the central nervous system and eventually the brain, or spinal cord and then the brain, they converge in one neural pathway. So your brain assumes the visceral pain is coming from the skin, and the brain cannot determine if it's coming from the skin, muscle, or if it's coming from an organ, if they're all converging. So heart pain felt in the shoulder or arm, because both send pain input to spinal cord segments T1 or thoracic 1 to thoracic 5. Another thing to look at is projections pathway for pain. So in your neck and shoulder, liver and gallbladder on the left side. On the right side, lung and diaphragm. Heart, that's your referred pain over there in your arm. But always on the left side. That's left side. That's right. You also have pancreas, stomach. That's not really where your stomach is all the way here. It's kind of over here, just below that. Small intestines, even though your small intestines is all the way up here and all around. Your kidney is very strange because it's on the side of your leg. So by knowing all these different things and knowing all these different parts in nursing will help you be able to say, do we need an MRI of an organ, or is this actual just pain on the skin? You can also have chemical senses, and when we talk about chemical senses, taste and smell receptors, because taste and smell are chemical. We're going to describe the receptors and the projection pathways for these two senses. So where did the pathway of taste go to the brain, and where did the pathway for smell go into the brain? Let's start with taste. So gustation or taste, sensation that results from action of chemicals of taste buds. 4,000 taste buds, mainly on the tongue. Some of them are going to be inside the cheek, soft palate, pharynx, and epiglottis. So sometimes you might breathe in something, or you might have some type of snot that goes back into the back of your throat that sounds weird, and you'll be able to taste it, because in your soft palate, pharynx, and epiglottis, you're going to have some taste buds. You have some main type of lingual papilla. Filiform, no taste buds. But filiform papilla are very important for food texture. Folate, no taste buds. They're not strong. They're very weakly developed in humans. Folate, if you look at your tongue, they kind of look like stretch marks on the side of your tongue. Filiform look like little hairs sticking up on your tongue. Fungiforms are the ones you kind of see, and you're like, oh, those look like little mushrooms sitting on the top of my tongue. So at the tips and sides of the tongue, that's where you're going to have all the little mushrooms. And the valate or circumvallate form a V in the back of your tongue, and this is going to contain about 1/2 of all the taste buds. So in taste, and you're going to kind of see in smell too, to actually be taste, molecules must dissolve in saliva and flood the taste pore. So taste actually has to touch the pore. You have 5 primary sensations. Salty. Salty is important because you might need to have ions. You might need electrolytes in your body. So if you're craving something salty, you're pretty much craving sodium chloride to go into your cells. Sweet. Carbohydrates and other foods with high caloric value. If you're craving sweet, you want glucose. You want energy. Sour, you can have acids like vitamin C and citrus fruits. Sour and also bitter can be off-putting for some people, and those can actually be the food is bad or something like that. But sour, acid, and other types of citrus fruits can be beneficial. Most of the bitter is going to be bad for you, like nicotine, caffeine, quinine, and morphine. These are all going to be types of bitter responses. And then probably about 30 years ago, they came up with umami, even though it kind of made sense, and Asian cultures have been talking about this for much longer than that. Umami is that meaty taste of amino acids. So if you want amino acids, you want to build protein, you want that umami flavor. More on taste. Taste is influenced by food texture, aroma, temperature, and appearance. So this means that not only are you going to feel that mouthfeel, you're also going to be pairing it with smell. So taste and smell is usually going to be talked about at the same time. One thing that students kind of get confused, hot pepper. So if you like spicy food. Spicy food and hot pepper do not go to taste buds. What they do is they stimulate the free nerve endings. So this is pain, not taste buds. And a lot of people, they like spicy food because spicy food is going to be a pain. That's going to be dopamine. That's going to give you that kind of nice feeling that you might not have had if you didn't have hot pepper. So that's going to be a pain, not taste. And also, there's going to be differences in sensation on the tongue. So tip, sweet, edges, salt, sour, and the rear is bitter. Not important for you to memorize, but if you do take sugar, put it on the back of your tongue, you will see that it doesn't taste as sweet as if it was in the front of your tongue. Taste is going to be controlled by three cranial nerves: facial nerve, glossopharyngeal, and vagus. Facial nerve collects sensory information from the taste buds over the anterior two-thirds of the tongue. So, front of the tongue, facial. Back of the tongue, the 1/3 is glossopharyngeal. And vagus, palate, pharynx, epiglottis. So by memorizing which one of these nerves is picking up this taste information, you can come up with more ways to diagnose brain injuries and other types of cranial nerve injuries, because if you put something on the front of their tongue, what does that taste like? And they can't tell what it is, part of their facial nerve might be damaged. So it's important to know front 2/3 facial, rear 1/3 is going to be glossopharyngeal, and then pretty much in the throat will be your vagus nerve. All the fibers reach solitary nucleus in the medulla oblongata. So all these fibers go to a nuclei called the solitary nucleus, and it's going to be in the medulla oblongata. From there, the signals are sent to two destinations: hypothalamus and amygdala. They're going to control the autonomic reflexes, salivation, gagging, and vomiting. And the thalamus is going to relay signals to the postcentral gyrus of the cerebellum for conscious sense of taste. So post-central gyri, that's behind the central sulcus. So then it's sent to the orbital frontal cortex to be integrated with signals from the nose, the eyes, form impressions of flavor and palatability of food. So in your cortex, that's when you start putting everything together, where you have a meal that looks good, smells good, and tastes good. That's going to be stored into a memory of something that you enjoy and probably want again. Now for smell, that's olfaction, and that's the sense of smell. Olfaction mucosa contains about 10 to 20 million olfactory cells. These are going to be neurons. They're going to have supporting cells. There's going to be mucosa and mucus on top, and this is going to be in the superior nasal concha in the roof of the nose. We don't have the actual picture for the bulb, but what it kind of looks like, you just have a little bulb. You have some fibers that hang down, and then there's a tract that goes back to the brain. Certain type of olfactory cells. These are going to be neurons. They're going to use chemoreceptors. They're shaped like little bowling pins. The head bears 10 to 20 cilia called olfactory hairs. Pheromones and other smells will stimulate them. So if you're kind of thinking, these little hairs, things have to pass by them. So when you breathe in, take a deep breath. These little chemicals in the air have to pass by these little hairs. Those hairs can then stimulate the nerves, and that's how we get the sense of smell. And then these are the only neurons in the body directly exposed to the external environment. They have a lifespan of only 60 days. The reason why is because they get worn out. They actually have to combat the external environment and not just be internally at all times. The basal cells continually divide and differentiate into new olfactory cells. They have supporting cells, and the basal cells from like 2 slides ago divide and differentiate to replace the olfactory cells. So action potential will then travel to the brain. You remember when we talked about phasic versus tonic. You remember that example of perfume or cologne before you go out. The olfactory receptors adapt quickly and then slowly stop sending signals to the brain. because sense of smell in your body is only designed to sense new smells and new dangers for yourself. Some odorants act as nociceptors on the trigeminal nerve, and this is going to be pain, ammonia, menthol, chlorine, capsaicin of hot peppers. The reason why these are going to be pain is ammonia, menthol, and chlorine, and even hot pepper can be dangerous for some people. So you smell it, you almost have almost not really a pain response, but more of an alert response to those types of smells, because those are going to be pain receptors and some smell. So you have tufted and mitral cells, axons from olfactory tracts. They reach the primary olfactory cortex, and that's going to be found in the inferior surface of the temporal lobe. So it goes to the primary olfactory cortex. It goes to the inferior surface of the temporal lobe. Secondary destinations: hippocampus, amygdala, hypothalamus, insula, and orbital frontal cortex. They identify odors, integrate smell with taste, perceive flavor, invoke memories of emotional response and visceral reactions. Fibers reach back to the olfactory bulbs where granular cells inhibit the mitral and tuft cells, and this is the reason why odors change under different conditions. Food smells more appetizing when we're hungry because there's going to be an external control on smell. Now, the next slide, again, isn't going to have a picture, but the main goal is the sense of smell does not go through the thalamus. It goes to all these other different parts of the brain, but it's one of the only senses that we have that does not go through the thalamus. And this would show you where it's going to, but you can see right here, none of those is the thalamus. So sense of smell does not go through the thalamus. And that concludes part 1.